a lot of different companies producing and formulating. Actually, the formulation is the most important things in this case. So they can formulate fungi and nematodes and virus, bacteria, or whatever to fight with arthropods. And it is the same case for disease. And also, it is really important thing that you can use as essentials. Is there any question? No. We can use these things for against the disease and against nematodes, against mollusks, and against weeds. So you can use different alternative to get good crop protection. So as I say before, the important things that the formulation or thinking methods and understanding the plant, understanding the past, actually it is one of the most important. Augustine gave some kind of information about the mealybugs taxonomy. For example, the first things, you have to know the plant very well, and you have to know which is the real past. And the taxonomy is really important. If you know the biology of the plant, of the past, or arthropod, or disease, or nematode, you can find best solution indeed. How you can use to crop protection, plant extract, bacterium, fungi, virus, nematodes, and the other things. But you have to, as I told you before, you have to know which is the species and which method is good for us. So from this point, I would like to give you some 3 examples for the new generation of crop protection. One of them are plant essential oils. So recently, it is really popular in the world. One of them is, so I'm just giving one example for each application or for each method. But plant essential is coming very well. For example, this is orange oil, but there are a lot of plant essential which is really good for crop protection. Clove oil, eucalyptus oil, mint oil, and cinnamon oil, and thymus oil. It's really having really big potential against the insects. I'm just talking about the insects. Of course, they are also important to control fungi. So I don't know what is your opinion about that, but so my impression is these kind of things are really important on chitin tissue in the environment. I'm saying in the environment because chitin is everywhere. And for example, chitin is on insects, very important part, and chitin is on fungi. It is also a very important part, and chitin is everywhere. And these guys are really, these elements, this formulation or whatever, are really important on chitin. So I'm just trying to say that, so these plant essentials are coming really good alternative for crop protections. First, I would like to say that. So I'm just giving a special example for orange oil. So, for example, orange oil, this is limonene. And so you can make a, different formulation. And what is the advantage on the plant protection? For example, you can use this kind of alternative essentials in organic use because there is no post-harvesting time, because it's pre-harvest. You can use this kind of things and you can harvest and you can eat, and they are not harmful on beneficial insects, predator insects and bees, and can be used spring tank and partner with, so you can mix this kind of essentials with even chemicals. What is the advantages you can use with the chemicals? It means that so you can even reduce the chemicals ratio if you use together with the essential. This is the other things, and there is no residue. And how they are affecting insects, mode of action. So first, they are infecting. So they in general, for example, so again, I'm talking about the orange oil. So they are just affecting the chitin and destroying the chitin cells, proteins, and the other things, and they are making some holes on the integument, and in this case, it has contact mode of action at the beginning, and the other things, they are also feeling the stigma, and the insect cannot breathe, and they are dying directly. Contact effect is really important for this kind of essential oils. This is different from the other oils which we are using for winter spraying and summer spraying. So it is coming from petrol or something like that. But these oils, these essentials are quite natural, and it has really important effect. And as I assume that, so this kind of essentials will be really important in future with the other things. So for example, they are using nanotechnology, they are using the other things, and people are working on these essentials, and in future it will be important. One of the most important problem of these things, they have some kind of phytotoxicity if you are using high ratio. But the mode of action is something like that. Look at this. So on mealybugs, so after 15 minutes, the spraying, and you can see that, so there is some kind of deformation on integument, and now after two days, So totally, because the insect is losing water if you are affecting the integument, and so they are just getting dry. And it is the same effect with aphids. And after 21, so after 1 day, the insects, aphids are getting dry after 1 day. So this is the one thing what I would like to say, that the essentials are coming for the next generation plant protection. And of course, we have a lot of things about the last years. We are getting a lot of improvements for the molecular studies. So at the beginning, so GMOs were really popular in New World, in America, and in Brazil, something like that. So we are just thinking that genetically modified organisms will solve everything. So we put in the plant DT, and we were thinking that everything will be okay, the plant will be great. But we at the end of the day, we got a lot of problem because of this technology, and we were just putting a lot of genes, and we were just putting genes with a lot of antibiotics, and we have some problem. And for this reason, this kind of GMO was not suitable and was not accepted, for example, in Europe. So it is not acceptable plants, and we are not using. So, but the genetical studies are moving on and getting a lot of advantage to have some kind of plant protection studies. So what I would like to say that, so now we have a new technology that we can silence the genes, for example, antennal organ, for example, ovipositor, for example. So they cannot find the plant, they cannot, so I'm just talking about the insect for sure. And so they cannot feed, so we can silence some kind of part of, some part of genes that they cannot use that ability to find, for example, plant, for example, they cannot lay eggs or something like that. So this is RNA based treatments. So this technique, so last 10 or 20 years, so we can make some kind of double strain RNA or DNA treatment, and we can get good results, and which will be really interesting because it will be really specific, it will be really target specific, and even gender specific. So this is really, really completely Can you hear me?
Yes, it's now
So
audible.
if
Yeah.
I'm talking too much, you can
No,
cut me
just
anytime.
a few minutes.
Few minutes, okay.
Yeah.
So this is RNA based treatment, and so now we can make different treatments on the plants. Even we can use the DS RNA to have some kind of application. And the other things, semi-chemicals, we have to talk about for next generation plant protection. And pheromone, hormone, and the other things are really important. So the insects are having communication with always with the chemicals, but we can use these chemicals for plant protection as well. How we are using. So this is the specific things that we can make some we can use them for monitoring, and we can use them for mass trapping or whatever. But on the other hand, we have a different idea, we have a different perspective that we can use these chemicals to attract natural enemies to all crops, and we can have more population of the natural enemies against the pests. And there is a really good example. For example, we can use these chemicals to attract Chrysoperla carnea or Neuroptera against aphids, for example, and we can have more population of the Chrysoperla carnea in the field, and we can fight with the aphids very well. It is the same for the parasitoids. We can use the pheromone, attract some parasitoids into the field to have nice, better biological control. Thank you for listening.
Thank you, Prof. Kaidan. Kita mempunyai waktu 30 menit untuk diskusi. Pak Agustin dan silahkan menjawab. We have 30 minutes for discussion on this section, and I don't see the question in the chatting for. Oh, ada. I have one question for you, Prof. Kaidan.
Okay.
Yeah, in the chat you can see the chat, and I think I will give first chance for you to answer. Okay, now, please. Nanti. Yang mana? Yeah, the first question. Mana? Ini ya. Show. Dari mana? Ya, oke. Iya. Dari mana? Yang mana nih? Sebentar, yang mana? Aswana. Bentar. Bentar. Yang di atas ya? Haman. Yeah, one question you can answer. How could we eliminate the wax layer? For millipede control.
Also, wax layer. So for that's a good question. Thank you very much for the question.
Yeah.
For example, these essential oils can you hear me?
Yeah. Can you?
Yeah. So this essential is really good for cleaning the wax.
Yeah.
And soup, for example, as well. So the other chemicals.
Mana apa pertanyaannya? Hah? Apa? Okay. Okay, one more question. Why is mealybug often causing problem for agriculture product export, for exporting agriculture products?
Again, please.
Why is mealybugs often causing problem in exporting agricultural products?
Actually, I don't know.
Yeah, the question is the mealybug
Because
sometimes
these guys are really so everywhere, and they can be everywhere.
yeah,
But
how important is
generally-
mealybug causing problem for agriculture product export?
You know, there are a lot of reasons, I think. So they have really hard to find on the products. So it's really hard to see them if they are really small, and they are always choosing the hiding place, so dark place. That is the reason that so they are always, you know, between the leaf sheets and something like that. So they are always hiding inside the hiding place, and they are trying to find a dark place. Maybe it's really hard to find, and after that, so it's a big problem. But I don't know how they are going everywhere. They are everywhere.
Yeah, the last question from Dr. Henry Bustamam about biological control. So, what biological control effort can be done against mealybugs?
Okay, so first, of course, I just would like to give some kind of summary about how we are making plant protection, crop protection, and how we should try to find different aspects of the not only mealybugs, so for all the life. But biological control is different issue. So first, again, you have to know which is your insects or mealybugs. And the other things you have to find the source, where is the origin of the insects. And after that, you have to go there and you have to find the best natural enemies. Of course, we can use natural enemies very well, such as one of the best good example is Planococcus citri and Cryptalomus montrosieri and Leptomastix dactylopi. You can use these two against the Planococcus citri very well, and it is the same for all of the insects and all of the mealybugs, and every mealybugs has really effective natural enemies, especially parasitoids. One of my friends, taxonomist Surgenia, told me that the parasitoids are the best taxonomists in the world. And every mealybugs and every scale insects or every insect has special parasitoids which can be really effective.
Okay, I think that's the answer, Pak Hendri. The rest question I think to Dr. Agustin Sarkani, silahkan dijawab langsung karena udah ada di situ di chat ya. Dari ada berapa nih? Ada 5 kali ya. Jadi yang pertama dari Miftahuromah ya. Lalu dari Rostaman, lalu dari Lili Cosmic Arts, lalu dari Aswana dan dari Maman Hartaman. The next question for Agustin of Kaidan. So Agustin will answer in Indonesia.
Yeah, thank you. Pak Rostama, terima kasih Pak Rostama. Untuk pembuatan slide ini Pak Rostama sangat banyak metode. There are so many methods
Agustin,
for.
okay.
Oleh.
Yeah. I was thinking that you are just speaking Indonesian.
So the participant asked how to prepare the mealybugs slide in the microscopic methods. So
I cannot answer this. I have to show it.
Yeah. Okay, but I will try to describe in Indonesia, maybe for the short answer.
But every individual has different different ingredients, and you have to use sometimes 1 day, sometimes 2 days, sometimes 3 days. So every individual has some kind of different style, so you have to work on it. And so really it is not easy to explain. So there is a procedure, but this procedures depends on the specimens. You have to change it. So it is not possible to explain online.
Ya. Oke, Pak Rustama, jadi metodenya banyak sekali Pak ini modelnya. Dan bahkan ada yang sederhana Pak, itu dengan cukup merendamnya dengan KOH, kemudian membersihkannya dengan alkohol, merendamnya ulang ke minyak cengkeh, dan kemudian taruh di balsem. Itu udah cukup juga. Tapi memang cara yang digunakan oleh kami di sini menggunakan metode Kostarab dan Kozat, ya, itu dengan menggunakan pewarna sehingga bisa lebih warnanya akan lebih terang gitu bagian-bagian itu. So Hojam, is there any simple way to making the preparates? Maybe
No.
just using the caranfil oils,
No, not really. So we have to follow the procedure. That's a really big problem.
Ya.
It's really
Katanya walaupun
long
itu,
procedure.
ya. Jadi walaupun ada yang sederhana, tapi untuk penyimpanan, untuk untuk tujuan identifikasi lebih jauh, memang menggunakan standar prosedur yang baku. Dan caranya sangat banyak ini, Pak Rustaman. Saya kira ada yang simple, ada yang sangat sedikit complicated gitu. Tapi hasilnya itu lagi-lagi tergantung tujuan kita. Kalau hanya untuk identifikasi sesaat, mungkin cara yang sederhana itu bisa dilakukan.
Agustin,
Yes,
it
sir?
is not complicated. It is hard procedure.
Sorry. Okay. The next question. Yeah. For the Dismisscoccus bonensis, it is the main pest of the sugar canes, Saccharum officinalis. And now already, I think not now, tidak sekarang, tapi sudah lama ini Pak. Itu direkam ada di Sumatera, terutama di Sumatera Barat, di perkebunan Sumatera Barat. Memang dahulu Sumatera Barat menjadi tempat perkebunan tebu ya, tebu yang cukup besar sebenarnya, tapi sekarang aja sudah mulai tidak ada lagi. So the Dismisscoccus bonensis is already now in Indonesia, in Sumatera.
Good for you.
Ya. So itu Pak Lili ya, Pak Lili. Kemudian Pak Azwana tadi sudah disampaikan oleh Borakaidan tentang kontrol yang menghilangkan lilin ya. Dan the most important things for controlling the mealybugs, untuk mengendalikan mealybug itu juga melihat waktu yang rentan untuk pengendalian. Jadi ninfa instar pertama itu dia crawler. Jadi dia bergerak aktif dan berpindah-pindah. Dan dia belum memiliki lilin. Jadi yang ini mungkin salah satu cara untuk pengendalian, waktu pengendalian. The time for the controlling is also important to control the mealybugs, right? So,
is it a question or you already answered?
Yeah.
The answer.
Okay. And yang lainnya, Pak. Saya kira itu kali, Pak.
Lili Kosmihat.
Ah, ya.
Lili Kosmihat.
Coba.
Lili Kosmihat. Siapa itu?
Bisa disebutkan, Pak? Pertanyaan terakhir ya. Kemudian untuk Pak Edi Susilo itu yang di yang di ini Pak ya. Memang di sorghum kita belum punya data, tapi bukan berarti tidak ada, saya kira. Dia akan mungkin ada di Jatropha curcas itu di jeruk ya. Jeruk lagi di jarak pagar ini itu ada 7 spesies Pak. 7 spesies. Dismicoccus brevifax mungkin yang paling dominan. Ada juga Planococcus citri. Ini bisa dilacak nanti Pak di scalenet.info ya. www ya. .scalenet.info. Nanti bisa dicari jenis spesiesnya, kemudian distribusinya, ekologinya ya. Host plan-nya juga disebutkan di situ. Itu sangat bisa dicari. Kemudian
Agustin, so
yes,
you are not asking, you are just answering the question.
Ini mana lagi Pak?
One small question for you, Prof. Kaidan. What is your main current research at this moment? I am interested with the information in.
Yeah, I saw that question. So I'm working on systematic entomology, plant protection. Systematic entomology, chemical control, biological control of the mealybugs. Mainly mealybugs.
Okay, that's I think answer the question.
Yeah, and molecular biology as well. Yeah. So I'm just doing molecular systematic as well.
Iya, mungkin nanti Pak Tarkus juga nanti bisa kontak-kontak kita Pak di Universitas Bengkulu. Kita juga sedang riset tentang mealybug ya, scale insect juga bersama Bora Kaidan. So I think for the next we will meet you in the next new collaboration with the other.
Of course, I will be really happy. So my university is really happy to contact with you. So Agustin, so we need the signature things. Did you send us? Yeah. For the make need collaboration. So for the collaboration with your university and my university, they were asking with the signature things and official things, and they would like to make a collaboration with your university. And thank you very much for inviting me in this meeting. And Agustin is a really good scientist, so he's always opening the new collaboration and new stage of the scientists. So sometimes I'm feeling myself that he's forcing me a lot, but it's really good things so he's doing good things. And thank you for the society and especially for Agustin, who is inviting me in this meeting.
Yeah. Thank you, Prof. Kaidan, Bora Kaidan. I think your presentation is very interesting. I think I will not conclude, but I will underline some important statement in the presentation. How we coping with increasing demand for foods in the future due to increasing human population, also environmental climate change, also decreasing arable lands. So we as who responsible for pest and disease control have responsibility to assurance that effort to increase agricultural productivity is assured by our effort to control pests and diseases sustainably. What I mean by sustainably is using technology that is based on natural products, whether from insects, plants, also from microorganisms, I think. That's, I think, what has been talked and discussed by us.
So, may I answer?
Also, Dr. Agustin talk as presenting important finding, and also we know that there are many species that had not been identified yet in this country, and this needs our concern for next research. Okay, the last thing, we will present certificate for Professor Burukaidan and Dr. Agustin.
Thank you very much.
Okay, please.
So thank you very much.
Okay,
for that.
yeah, Dr. Burukaidan. Okay, and also,
Thank you.
and Dr. Agustin sudah ya. Nah ini Dr. Agustin Sarkani. Okay, that's all for this session. I'm sorry for saya mohon maaf atas kekurangan ya, for the lack of presentation, my guiding. So waktu saya kembalikan to moderator, to host ya, to the host. Okay. Makasih. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Profesor Burukaidan. See you later.
Eyvallah.
Tersegar itu deh.
Are we done with the everything, Agustin?
Yes, sir. So you can leave. The next is the parallel section. So we have more than 43 presenter. But if you want to join in the parallel section, we will send you the new link. So
okay, I'm fine with this.
Thank you. So we have the seminar.
I have to go to school right
maybe,
now.
for 4 hours later for the presenting some research article in our country. So thank you very much, Professor Burukaidan.
Thank you very much, Agustin.
We wish you will be safe and healthy, and we hope everything will be good in the next. Thank you.
Okay. Thank you very much.
Thank you.
Thank you very much for everyone.
Thank you. Thank you very much. See you.
Bye-bye. Bye-bye.
Bye. Bye-bye.
Terima kasih kepada Profesor Dwinardi Aprianto yang sudah memandu sesi entomologi. Dengan telah berakhir sesi entomologi, maka berakhir pula sesi materi untuk acara seminar nasional virtual pada hari ini. Setelah ini, kita akan langsung memasuki sesi paralel yang akan dimulai pada pukul 13.00 waktu Indonesia Barat.
Thank you to Mr. Dwinardi Aprianto who had conducted the entomological session. The entomological session has ended, and now we will move to the parallel session, which will begin at 1 p.m. West Indonesian time.
Baik, sebelum berakhir, kami mohon untuk Bapak, Ibu, dan seluruh peserta agar dapat mengaktifkan kamera masing-masing. Kita akan memasuki sesi dokumentasi. Kami persilahkan. Baik, kita mulai. Untuk slide pertama, kami persilahkan. 1, 2, 3. Oke, slide selanjutnya, slide kedua. 1, 2, 3. Oke, slide selanjutnya. 1, 2, 3. Oke. Slide selanjutnya. 1, 2, 3. Oke, terima kasih. Kami sampaikan kepada seluruh pemakalah atau presenter, sudah tersedia 7 room yang kami persilahkan untuk seluruh presenter memasuki room masing-masing sesuai dengan yang sudah ditentukan oleh panitia. Kami sampaikan untuk room pertama bidang perlintahan virus, untuk room kedua dan ketiga perlintahan bidang hama, untuk room keempat perlintahan bidang cendawan, untuk room kelima perlintahan bidang bakteri, untuk room ke-6 bidang agroekoteknologi, ilmu tanah dan biologi pertanian, dan untuk room terakhir perlintahan bidang hama dan virus. Kami mohon untuk seluruh pemakalah dan presenter agar dapat stay di room yang sudah ditentukan 5 menit sebelum acara atau sesi paralel dimulai. Dan untuk seluruh peserta, kami ucapkan terima kasih sudah berpartisipasi hingga sesi entomologi selesai. Bagi seluruh peserta yang ingin mengikuti sesi room paralel, kami persilahkan untuk memasuki room yang sudah tersedia. Kami ucapkan terima kasih sekali lagi. Saat ini kita akan memasuki sesi istirahat sampai berjumpa di pukul 13.00 waktu Indonesia Barat. Selamat beristirahat.